Assalamu alaikum wa rahmatullahi wa barakatuh. Hello everyone. Today inshallah we will complete explanation of module 102 inshallah module blood. Inshallah we will start today with platelet or thrombocytes. Inshallah let's get started. So platelets. The characteristics of platelets is it is a small oval granulated and non-nucleated cells. Okay, it is non-nucleated. It is like red blood cells, red blood corpuscle. So it is radiation 2 to 4 micron. Okay, let's count 300,000 millimeter power to 3. It is lifespan 8 days. Okay, this is an important MCQ information. So let's move to the steps of production of platelets. Okay, let's start from pluripotent uncommitted stem cell by interleukins will become myeloid stem cells. Myeloid stem cells, okay, turn to monocytes and macrophage, neutrophils, basophils, xenophils, and erythrocytes. Okay, and megakaryocytes, but under proper stimulation. If you notice, mainly all of these cells are immunity cells, like macrophage, neutrophils, inflammation cells. So myeloid stem cells, okay, by thrombopoietin, which produced by liver and kidneys, okay, became megakaryocytes. It is a large cell from its name. Okay, 70% of platelets, it is from bone marrow. Started the site of formation, it's in bone marrow, as we said and mentioned before. 30% is stored in the spleen. Okay, in splenectomy procedures there is increase in platelet count. Why? Because the 30% is not stored in spleen, it just become added to the 70% in the blood. Now let's go to the structure of the platelets. The platelets membrane, it's coated by glycoprotein coat. Okay, why? Because to prevent adherence to normal endothelium, to not make thrombocyte or thrombosis in normal cases. Yes, it just adherent to injured endothelium. If you notice, this is for example capillary. Okay, the normal capillary platelets doesn't stuck on it. If there are injury in capillary like here, platelets came and adherent in this site to close the foramen. Okay, so the coat contain many receptors for collagen and for von Willebrand factor. Okay, you can just call it VWF. Okay, also contain phospholipids. Okay, the membrane forming invagination, open canalicular system, pathway for uptake calcium and release of intercellular substance. Now let's move to the cytoplasm. Inside the cell, it is contain mitochondria, microtubules, Golgi apparatus, lysosomes. That's it. And also contain contractile proteins, actin and myosin, to help it move and to stuck to the site of injury, like this. Make a podocyte. like this. Okay. And also contain granules. We will discuss this later, inshallah. Now let's move to another subject, hemostasis. Hemostasis, as we said before, it is the prevention of blood loss after injury. Prevention of blood loss. In cases of hemorrhage, trauma, any case of blood loss. Okay, hemostasis, it's a mechanism to help prevention of blood loss. Let's start by local vasoconstriction. Vasoconstriction mean this is normal capillary, okay, normal size capillary. When vasoconstriction became smaller like this, smaller in diameter. Yes, that's an injury site. Secondly, formation of temporary plug, hemostatic plug, okay, platelet reaction. Okay, if your tip of finger, okay, get injured by something small like piece of wood, glass, something like this, okay, very small opening, it's just make a plug, a small platelets to close the injured site. If it is large, like big wounds, it will formation clot. Okay, that means it's called this procedure is called blood coagulation. Okay. If the injury is just healed, okay, we must remove the clot. It's removed by dissolution of clot, okay, by substance. Inshallah, we'll discuss it, inshallah, specifically from plasmin, etc. Like this. Okay, now let's just discuss it specifically. Firstly, let's discuss the local vasoconstriction. It is immediate reaction, immediate reflex. Okay? Due to, firstly, nervous reflex. Okay? By nervous action. It starts by pain sensation from traumatized vessel. Yes. Then local myogenic contraction. Thirdly, by humoral or chemical substance like ADP, serotonin, thromboxane A2, okay, released from platelets. So thromboxane A2 is released from platelets to make vasoconstriction. As we said before, vasoconstriction, that means the normal sized capillary turn into small sized capillary to prevent blood loss. Secondly, let's go to formation of temporary hemostatic plug or platelet reaction. This mechanism specifically work in small injury. It starts by platelet adhesion. For example, let's imagine this is the normal blood capillary, okay, get injured, okay, in this site. So the blood start going outside the capillary. The normal platelets in the blood just go and stuck in this site and make a plug so that can prevent blood loss. Specifically, mechanism is firstly platelet adhesion by receptors to exposed subendothelial collagen and this site endothelium, okay, become clear. Yes, and von Willebrand factor. Von Willebrand factor is a glycoprotein also present in plasma and bound to factor number 8. Factor 8. So, if someone asks us about factor number 8, just tell him that it is bound to von Willebrand factor. Secondly, platelet activation after adhesion. After adhesion, platelets are activate each other. Okay? It become swell and it change in shape. And release pseudopodia. This is normal. Platelets became like this. Okay, normal pseudopodia to stuck in the site of injury and release granules. Activation is increased by ADP and thrombin. Okay. Thirdly, platelets release reaction. It is calcium dependent reaction. Okay, there is need of calcium in this reaction. It release of dense granules and alpha granules. Dense granules is non-protein like ADP. Make activation and aggregation, fusion of platelets. And serotonin. Maintain vasoconstriction and reinforce. And also calcium. So dense granules, it is non-protein. It doesn't contain protein. Alpha granules, it contains protein. So alpha granules, as we said, contain protein. It's contain clotting factor, like factor number 13. It's stabilization of fibrin clot. And platelet derivative growth factor, or PDGF. It stimulates growth of endothelium and smooth muscle. And platelet activating factor, PAF. It helps platelets aggregation. So, that help platelet aggregation, as we said before, it is like PAF or platelet activating factor and ADP and thromboxane A2. Okay. it can be act like MCQ question. Okay. Like mention one of the following is not platelet activating factor or aggregation factor. You can just throw these answers out and choose the left. This is mechanism of working. Membrane phospholipids like PAF, platelet activating factor, start with arachnoid acid, cyclooxygenase, make prostaglandin, started prostacyclin synthesis and thromboxane synthesis. Prostacyclin synthesis of healthy endothelium and thromboxane synthesis of platelets. Thromboxane A2 is increased in calcium, increased free calcium in platelets. Thromboxane A2. It started vasoconstriction and platelet release and increased platelet aggregation. On the other hand, prostacyclin make the opposite, vasodilatation and decrease platelet release and decrease platelet aggregation. So prostacyclin is the opposite thromboxane A2. Let's keep platelet plug localized to site of injury and prevent clot spread to healthy area. Okay. Medicine like or a drug like aspirin inhibit COX formation. COX means cyclooxygenase. So prevent thromboxane A2 and prevent prostacyclin. Prevent both of them. So aspirin is a good drug to prevent thrombosis, to prevent emboli. So endothelial cell can produce new cooks within hours. Okay, platelets cannot. Okay, daily intake of aspirin decrease clot formation, prevent myocardial infarction. Myocardial infarction means that side of the heart is died because the loss of oxygen, because thrombus stuck inside the blood capillaries. Okay. Fourth point is platelet aggregation, as we said before, by ADP and thromboxane A2 and platelet aggregation factor. Increase the aggregation and make more activation of the platelets. More release of granules, self-propagating process, and in platelet plug formation. The next point is platelet procoagulation activity. Platelet factor 3, PF3, exposed on platelet membrane, provide ideal surface for concentration and activation of clotting factor. Okay. Platelet acts as catalysts for coagulation. Yes. Final point is platelet irreversible fusion by ADP and thrombin platelet enzyme. Okay. Now let's move to another point or another subject, blood coagulation, blood clotting. Let's discuss it specifically. It is by clotting factor, okay, plasma protein, mainly beta globulins synthesized by liver. If you remember, we have studied alpha and beta globulins and gamma globulins. Gamma is for immunity, and alpha and beta act as a carriers. Okay. Now some of beta, mainly beta globulins, okay, it's act as a blood coagulant protein. Yes, proteolytic inactive enzymes, when activated, activate other, okay, cascade of reaction ending clot formation. That's mean, for example, factor 1 activate factor 2, activate factor 3, and this chain till make clot formation. There are two main pathways, intrinsic pathway and extrinsic pathway. Intrinsic pathway is contact with subendothelial collagen, okay, in vivo, and contact with negative charge wet surface in glass in vitro. So intrinsic pathway can act in vivo, also in vitro. Okay, this is important information. So let's start it. Factor number 12, okay, exerted by HMW, kininogens and kallikrein in plasma, turn to factor 12A. 12A activate factor 11. Factor 11A activate 9. So started 12 to 11 to 9. Then factor 9 form a complex with factor 8A activated by thrombin, okay, separated from von Willebrand factor, activate factor 10. So, as we said before, 9 with 8, 9 with 8, activate factor number 10. Factor 10A, in presence of phospholipids and calcium, and factor number 5, okay, factor number 5 is one of the requirements to start. Activate prothrombin into thrombin. Prothrombin into thrombin. If you remember, prothrombin was one of the plasma protein we mentioned before. So, thrombin converts soluble fibrinogen into insoluble fibrin monomer. Start to making clot. In these points, start making clot. So, factor 10 in presence of phospholipid and calcium, and factor number 5 start to convert prothrombin into thrombin. Okay, thrombin converts soluble fibrinogen into insoluble fibrin. Then, fibrin monomer polymerize, make a loose mesh of fibrin. Small net of fibrin. Loose mesh is converted into tight mesh by factor number 13. Factor number 13 is fibrin stabilizing factor. Okay, just stabilizer. Activated by thrombin, presence of calcium. Okay, the network became tight. Okay, to close the injured site. By contraction of actin and myosin of platelets, clot retraction, and squeeze the serum. Serum is devoid of prothrombin and fibrinogen, factor 5, 8, 13 consumed, and contain a large number of serotonin. Okay. Net is a formation of a clot, as we said before. Fibrin thread by platelets, blood cells, and plasma interrupted in the clot. Calcium is required for all reaction except first two steps. This is a very important MCQ information. So first two steps, there is no need for calcium. Yes. That was the intrinsic pathway. And this can be in vivo and in vitro, inside the body and outside the body. Let's move to extrinsic pathway. Damaged tissue just in vivo. Just in vivo. Just inside the body, not outside the body. It starts by release of thromboplastin. Okay, TBL and factor number 3. Activate factor number 7. Number 7A, factor number 7A is in presence of phospholipid, TBL and calcium. Activate factor number 9 and factor number 10. Factor number 10A, okay, complete the same mechanism as we discussed it before. Interaction between two systems. In the body, following injury, okay, clotting initiated by both systems, intrinsic pathway and extrinsic pathway. Okay, extrinsic system is rapid because there is no steps as the intrinsic factor, extrinsic pathway. So it just take 15 seconds. But intrinsic system is slow, take 1 to 6 minutes. extrinsic system or extrinsic pathway leads to formation of thrombin rapidly, okay, because less in time, and activates factor number 8 and 5, leading to activation of intrinsic pathway. Factor 7A, which is extrinsic pathway, activate factor number 9 in intrinsic factor, and factor number 10, which is common. So extrinsic factor help in activation of intrinsic factor, because a lot of reasons. It is less in time, it is more rapidly, it is activates factor number 9 and factor number 10. Action of thrombin, okay, it is important. Activates fibrinogen to fibrin, and activate fibrinogen group 5, 8 and 13. Okay, it's accelerate platelet segregation and accelerate formation of more thrombin from prothrombin. Yes, more thrombin, it's positive feedback. Okay, positive feedback means more thrombin, okay, help to release more thrombin, like this. So there is increased thrombin in all steps. Okay, let's go anti-clotting mechanism, limiting reaction. The aim of this mechanism is prevent blood clotting in healthy blood vessel and breakdown of any already formed clot. It's in cases of myocardial infarction, any stroke, like this. In general, limiting reaction, smooth vascular endothelium, okay, prevent activation of platelets and factor number 12. Okay, in smooth, in normal, not in injury, okay. And also rapid blood flow. So the blood must be fast, going in specific speed. Yes. And removal of activating clotting factor. Also heparin. Heparin is a natural anticoagulant. Okay. Now let's move to specific limiting reaction. Thromboxane A2 and prostacyclin. As you said, they are antagonized each other. So prostacyclin prevent the mechanism work of thromboxane A2. Also antithrombin 3. Antithrombin, from its name, antithrombin, prevent thrombin. Binds and inhibit activity of factor 9 and 10 and 11 and 12. Okay, bind facilitated by heparin. So we have to get back to heparin again. That make us know that heparin is the main drug to prevent clot formation. Also fibrinolytic system, endothelial cell, okay, express thrombomodulin, became thrombin, okay, by action of protein C, activate protein C, make factor 8A inactive and factor 5A inactive. Okay. So inactivates inhibitor of tissue plasminogen activator, turn plasminogen into plasmin, and plasmin make lysis fibrin. Plasmin main action is lysis the fibrin, is dissolve this network. Okay, dissolve it. Now plasmin, which name is fibrinolysin, from it fibrin, fibrin, and lysin means breakdown. So it is lysis fibrin and fibrinogen. Fibrinogen degradation products, okay, inhibit thrombin. Okay, Streptokinase and urokinase, it is a bacterial enzyme in human TBA produced by rDNA technique even soon after onset of myocardial infarction. Within 6 it is not important information, just for reading. That was our lesson today. Thank you for listening. Have a nice day and good luck.